ناخذ نص ساعة نتكلم على different repair نص ساعة different repair mechanisms. So DNA must be protected. حكينا على واحدة من الأشياء اللي ممكن تصير أو one of the mechanisms by which DNA is preserved هي بالDNA polymerase نفسه إنه عندنا proofreading mechanism وعندنا ability of the DNA polymerase to add the right substrate and so on. طيب. لكن افرض إنه صار في عندي damage in the DNA. Afterwards, how can the cell fix the problem or the mutation? There are different mechanisms. A long list of mechanisms. We'll take them one at a time and simplify the different mechanisms. أول شيء الوقاية خير من العلاج، صحيح؟ أو قنطار وقاية خير من العلاج or whatever. Okay. So the idea, if there is something damaging to the DNA, let's remove it, eliminate it altogether قبل ما يعمل أي شيء. لين يدخلوا على البيت ويعمل errors or whatever, يعمل فوضى. So the cell has a certain mechanism, enzymatic mechanisms, like superoxide dismutase and catalase. So what they do, that superoxide is a free radical. A free radical, it's missing an electron. It's missing an electron. بده electron من أي حدا بروح بيهاجم أي molecule, including DNA, taking an electron from this molecule. If an electron is removed from DNA, that can lead to mutations. That can lead to something like double-strand break in the DNA. Okay? It's a crucial bug. So this free radical can be converted to hydrogen peroxide by superoxide dismutase. Hydrogen peroxide is also a reactive oxygen species. Although it is not as damaging as superoxide, it's still reactive oxygen species and it must be removed. So there's another enzyme called catalase. It converts the hydrogen peroxide into water and oxygen molecules that are not damaging whatsoever. Okay. So let's say that there's damage that occurs on the DNA. How can it be reversed? Directly? Well, again, there are different mechanisms. One of them exists in bacteria only. If UV light hits DNA and you have two pyrimidines on the same strand, one after the other, you can have the formation of what is known as pyrimidine dimers. So the two pyrimidines would be cross-linked together, covalently. When the DNA polymerase comes in and when it tries to read the two T's, for example, the two pyrimidines, well, it cannot, of course. So they must be removed. Bacteria, and only bacteria, have an enzyme known as photolyase. What it does, it converts the covalent linkage between the two pyrimidines back to the normal condition. So it removes this cross-linking of pyrimidines. Okay? Altogether. In our cells, in case of methylguanine, we talked about methylguanine before, and we said methylguanine pairs with thymine instead of cytosine in DNA. Well, if methylguanine exists in DNA, we have an enzyme known as methylguanine methyltransferase. What it does is that it removes the methyl group from methylguanine, converting it back to normal guanine, and that's it. Problem fixed. Let's say that we have different types of errors. Let's say that the cell does not activate the methyltransferase. There are a number of mechanisms known as excision repair pathways. Excision يعني cut. Okay? What they do, in principle, is that wherever there is damage in the DNA, lesion, mismatch, let's say, the DNA is cut, creating nicks, N-I-C-K. The fragment of DNA is removed, only one strand that contains the lesion, and it can be and the gap is filled in by a DNA polymerase. So there are different types of excision repair pathways. One of them, the common one, known as a general excision repair mechanism, or known as nucleotide excision repair mechanism. So the idea is quite simple. عندنا pyrimidine dimer right here. Simply, you have this system activated, creating two nicks surrounding the lesion. This fragment is then removed by a helicase, and the gap is filled in by a DNA polymerase 1, followed by a DNA ligase, creating a phosphodiester bond. That's quite simple. In bacteria, we have different proteins known as UVRA, UVRB, UVRC. They form a complex known as UVRABC. Each one of them has a function. واحد منهم بيعمل recognition للدامج، الثاني بيعمل cut على اليمين، واحد بيعمل cut على الشمال، بيجي بعدين helicase, it removes this fragment, DNA polymerase 1 fills in the gap and DNA ligase and that's it. هذا موجود في البكتيريا. ماشي؟ هل هذا السيستم موجود عندنا in our own cells? Yes, we do. Yes, we do. And if this system is defective, we can have a condition known as xeroderma pigmentosum. Xeroderma pigmentosum is basically when you have defective repair mechanism, general excision repair mechanism, that makes cells sensitive to light, to UV light. So the DNA is damaged, but it cannot be repaired. Okay? We have a number of proteins known as XPA, OB, C, D, E, F, G. Each one of them has its own function. One of them recognizes the lesion, another is an endonuclease, a third is a helicase, a fourth is a supporting molecule, and so on. Each one of them has a certain function, but eventually what they do is the same idea, which is basically cut, remove, and fill in the gap, and that's it. ماشي؟ هلا. In addition to these proteins, XPA all the way to G, we have two other proteins that I would like to emphasize on. One of them is a transcription factor, known as transcription factor 2H. راح نحكي على transcription factor 2H later on. It functions as a helicase. It functions as a helicase. So it's the one responsible for removing the cleaved DNA fragment. Okay? Another one is known as replication protein A, RPA. Replication protein A is the same as the single-stranded DNA binding protein اللي بيعمل protection of DNA, the replication process, protecting the DNA from hairpin structures from being degraded and so on. And it's the same one. So what it does, it coats the single-stranded DNA, protecting it, and allowing for the DNA polymerase to fill in the gap. ماشي؟ إذا هاي البروتينز موجودة عندنا in our system. هلا you can have different types of xeroderma pigmentosum depending on which protein is damaged, which gene is mutated. لو عندنا mutation with XPA, that can lead to a certain type of xeroderma pigmentosum versus XPC and so on. Fine. Let's say that we have transcription. RNA polymerase is synthesizing RNA. It hits a lesion in a pyrimidine dimer, for example. RNA polymerase cannot read the DNA template. It stops. Once it stalls, once it stops, that sends a signal to different proteins. Hadoli proteins, they are recruited to where the lesion is. Again, it's repaired by an excision repair mechanism, and the RNA polymerase can continue with the RNA synthesis and transcription. The coupling of transcription and DNA repair is catalyzed by proteins known as CSP, as well as XPA, اللي اشتغل معنا قبل بالxeroderma pigmentosum, as well as these two that we talked about before as well. Okay? هلا, if you have a mutation in CSB protein, that can lead to a certain condition known as Cockayne's syndrome. I put in an illustration for an individual affected by such mutation and different features associated with this condition. I encourage you to look at Google images, look at real patients affected or having this condition right here, this syndrome. It's quite severe. Okay. So, but they do the same thing. They recognize the lesion, cut, and then fill in the gap, and that's it. Okay? That general excision pathway, it works on all types of mutations or lesions that occur in the DNA, but we have specific excision pathways. Remember last time I said that you can have uracil in DNA. امتى ممكن يصير في uracil in DNA? Mutation. What type of mutation? What type of mutation? Deamination. Deamination of what? يلا حذر فزر. طيب كيف ثايمين؟ 10 مرات قال لي ثايمين، أنا بضيف ساكت. يلا. Cytosine. Cytosine is deaminated. It can be converted to uracil. Okay? And the presence of uracil in DNA is abnormal. And it's recognized and fixed by certain enzymes known as DNA glycosylase. But these DNA glycosylases in general, what they do is they remove the abnormal base, creating AP sites. يعني apurinic أو apyrimidinic sites. السايتس اللي فيها الفاسفيت والشجر بس ما فيهاش base. Okay? Creating AP sites which can then be fixed. كيف? Here's an example. عندنا الuracil DNA glycosylase. It recognizes specifically, عشان هيك نحكي specific excision pathway, specifically they detect the presence of uracil in DNA, and الuracil is complementary to G, لأنه originally كان عندي إيش؟ GC. Deamination of C creates uracil. This is abnormal. بيجي DNA glycosylase. It removes the base, uracil. بعدين بيجي endonuclease. It makes a phosphodiester bond, eventually removing the phosphate and sugar. Here you have a gap. It's filled in by DNA polymerase, and complementary to G, you have C. Problem fixed. أمور تمام. حلو؟ أي سؤال؟ So far. Okay. Let's say that replication is done, and the DNA polymerase has made a mistake during replication. G حط إيش؟ T. صار عندي mismatch. Somehow صار عندي mistake. How can this be corrected? In bacteria, there is a system known as the Mutant system, kind of mutation. The Mutant system. It involves 3 proteins, Mut S, Mut L, and Mut H. They do the same exact thing. They recognize the lesion, they make a single cut, مش two, زي اللي حكينا عنه بالgeneral excision pathway, a single cut, followed by an exonuclease, cut, removing the strand that contains the lesion. Then DNA polymerase fills in the gap, DNA ligase, and that's it. حلينا المشكلة. That's in bacteria. هلا. فاحنا حكينا انه صار عندنا mismatch. نعم.
ممكن تعيد دكتور.
أي أوكي. ففي عندي بالبكتيريا لو عندي mismatch، عندي DNA replication والDNA polymerase عمل mistake، يعني قرأ الجي made a mistake حط T، فعندك GT pairing. That's not normal. That's a mismatch. بيجي الميوت system, mute L, S, and H. They recognize the mutation. Or the mismatch. They make a single cut. مشترين زي general excision pathway. Exonuclease removes the strand that contains the lesion. DNA polymerase 1 fills in the gap, and we're done. منيح خلاص هي هذه simple. هلا فأنا عندي بالDNA replication خلصنا عندي mismatch. أجا repair system أجوا هذول الميوت S و L و H طلعوا عليه بحكوا إيه عندي G وعندي T. Both of them are normal. يعني ما عندي U نحكي اليو هي الغلط. No, عندي G or T. Both of them are normal. How do cells know which one of them is the wrong base, or the base in the wrong place? The mechanism in bacteria is methylation. DNA is methylated at adenine by an enzyme known as adenine methylase. What it does, and no, and the two strands, and these two strands are methylated at adenine. When DNA is replicated, you have one strand that is methylated, and the other strand is not methylated. And when this system comes in and it sees that there's a lesion, it looks at the methylated strand and says, this is the original one. This is the original one. And the other strand is the new one that must be fixed. فبيعرف انه هو لازم انه يشيل التي. So again بيعملوا الشغل اللي هم بيعملوه. Okay? Does this system exist in our cells? We sure do. In humans, in human cells, we have a similar system. We have similar proteins. Specifically, we have two proteins, MSH and MLH, and they are homologous, the mute S and the mute L, bacteria. And these two proteins, they can recognize mismatches in replicating DNA, whether the mismatch exists in the leading strand or the lagging strand. Eventually, what they do is they make a cut, allowing for the DNA polymerase to start over, to synthesize, to continue synthesizing the leading strand, or to replace the part of the opposite fragment that is cleaved. And that's it. فبنعبي الفراغ and mismatches are removed and repaired. كويس؟ كيف؟ هلا ايش هو الميكانيزم؟ كيف بتعرف اي وحده هي الصح وايش الغلط؟ هلا if they recognize that replicating in the female mismatch وبشوفوا انه في عندنا DNA that is being replicated بيعرفوا ان هذه هي the new one. لكن افرض انه المسماتش صار بعدين او اكتشف بعدين بس خلص ال DNA replication. What is the mechanism? I don't think anyone knows ايش الميكانيزم اللي يعني مش زي البكتيريا. انه يعرفوا انه هذه هي the new one, هذه هي the old one. It's a different mechanism. I don't think it is known yet. It turns out that these two proteins can be mutated in bacteria. But if the repair pathway is defective, that leads to accumulation of mutations. The hallmark of cancer is this, accumulation of mutations. إنه cancer بيصير في عنده كتير mutations أو السيلز العادية مثلا لو صار عندها كتير mutations that can lead to cancer eventually. So it turns out, when these proteins are defective, they can lead to, or this can lead to a type of colon cancer known as hereditary non-polyposis colon cancer, which constitutes about 15% of colon cancer cases. The HNPCC. It's hereditary, so it can be inherited من parents to children. وحتى البارنتس إذا كان عندهم defective gene راح يكون عندهم prevalence of cancer عندهم راح يكون كثير عالي. هلا 50% من these 15% فيهم الميوتيشن بالميوت S. والremaining large portion of the remaining 50% they have the mutations in the MLH gene. فهلا بحاولوا يعملوا genetic testing للأشخاص عشان يعرفوا عندهم mutation بهذول ولا لا to protect these individuals. طيب DNA polymerase synthesizing DNA. أجا عند lesion, أجا عند pyrimidine dimer. It stops. Again, it's decision time بالنسبة للسامي. تموت حالها ولا continue with the DNA synthesis مع المستيك اللي موجودة. طبعا الروح غالية, so what cells do, they prefer to continue with DNA synthesis with a mistake, hoping that it can be fixed later on after replication is completed. So there's a mechanism that allows for the DNA polymerase to bypass the mutation, to jump over the mutation and continue with the DNA synthesis. Okay? So it's known as translation DNA synthesis. This mechanism allows the DNA polymerase. What it does basically, if we have a pyrimidine dimer right here, it allows the polymerase to insert, to add any base, okay, so that it can continue. Any base complementary to the pyrimidine dimer. So, for example, right here it adds GA and it continues. هلا هون لازم يكون عندي ايش؟ TA و TA. لكن it adds a G. That's a mismatch that can be repaired later on. طيب? So although the system is efficient in allowing cells to survive, it's efficient in allowing DNA polymerase to finish its job إلا إنه it is error prone. يعني there is high probability of introducing new errors in the DNA. ماشي؟ Any questions? Yes? مثلا اوكي اذا احنا عندنا DNA polymerase شاف انه في pyrimidine dimer. It stops. It cannot read this pyrimidine dimer. لكن the DNA synthesis is quick. It must be finished immediately. Or it must be finished fast. Otherwise cells die. Cells do not like to die طبعا. So this mechanism بيخلي DNA polymerase يحط أي base complementary للpyrimidine dimer و hoping إنه بس يخلص ال DNA replication إنه السيل راح تعمل scan لل DNA ولما تشوف إنه في errors it can be fixed. Okay? هذا الميكانيزم هذه أو الماشينري هذه allows DNA to add any bases. فأنا عندي pyrimidine dimer هون. Complementary to the pyrimidine dimer, any base is added, so you have addition of G, addition of A. Just random. Now, it should be here AA, and the cone TT. Okay? So let's say, it's continued. DNA replication is continued. It's done. As are different repair mechanisms, بعد the replication شافوا إنه عندنا mismatch. عندي T, G. هلا بدهم يعرفوا أي واحدة هي الصح، أي واحدة هي الغلط. They can remove the T, placing a C instead of it. صار عندي mutation. بدل ما يكون عندي AT صار عندي GC. Okay? So the system هذا is efficient. لأنه بيخلي ال DNA polymerase يخلص ال DNA synthesis وبيخلي السيل عايشة لفترة أطول. لكن it introduces mutations in the DNA. Quite simple. ماشي؟ آخر mechanism هو recombinational repair mechanism. Remember إنه إحنا diploid. Remember إنه أنا عندي 2 chromosomes 1. Chromosome 1 من الfather, chromosome 1 من المother. Let's say that you have a double strand break. The DNA itself is broken up into 2 pieces, 2 strands. We have 2 systems that take advantage. Well, we have 2 systems. One of them takes advantage of having a homologous chromosome. The system الأول, which is you can say a dirty system. سريع, efficient, لكن what it does basically, it takes the 2 strands and it puts them together. Okay, it ligates them together. هذا اسمه non-homologous end joining. So joining the ends of chromosomes that may not be homologous to each other. So non-homologous end joining. Now, the problem with this system, although it fixes the DNA, it creates mutations that as it attaches the strands together, some bases can be deleted in the chromosome. Okay? The other system is known as homologous recombination, which takes advantage of having a homologous chromosome. لازم الكلر يكون شوي حلوة على chromosomes is broken up. You have a double strand break. And we have a homologous chromosome. With homologous recombination, one of the strands is cut from the other good chromosome and it is placed in the other damaged chromosome. Okay? ماشي? So one good strand is cut out and placed on the other chromosome. Now you can have a DNA polymerase using the good strand to fill in the gap. وبالكروموسوم المنيح اللي اخذنا منه فراجمنت واحدة من الستراند الثانية نورمال ما فيها mutations ما فيها break. It can be used as a template to fill in the gap. So eventually you will have a chromosome where we've taken part of the other chromosome out when we put it in and we can fill in the gap in both of these chromosomes. So this is known as homologous recombination. Is that clear? واضح؟ نعم.
ممكن نعيدها؟
نعيدها. نعم. نعيدها. Non-homologous. Non-homologous end joining. It's just that if you have a chromosome that is cut, what it does, it takes both strands that are cleaved, it puts them back together, it glues them. Okay? The problem with this mechanism, a few bases can be deleted out, creating mutations. That's it. And with this mechanism right here, with the homologous recombination, the cell takes advantage of having another copy of a chromosome. So we have another copy of chromosome 1 or 2 or 3. So let's say one from the father, one from the mother. Let's say that one of them is damaged. Okay, and you have a double strand break or a missing strand, let's say. Well, it takes advantage of the other chromosome. It takes the same fragment, it aligns the two chromosomes together. بيصير في عندي exchange of genetic material. So what happens, we take part of this chromosome right here and we place it on the other chromosome. Now this chromosome contains one good strand. صح؟ لأنه أخذناها من الكروموسوم الثاني, the good one. So this chromosome has one good DNA fragment, so it can fill in the gap. And the other chromosome that is now also missing a fragment, and we took it out, we put it on the other chromosome, it has also one good strand. The gap can be filled in as well. لأنه عندها template. هذا هو أهم شيء، إنه يكون في عندي template. فالتمبليت تبعت هاي الأزرق أخذناه من الأحمر، من واحدة من الستراندز. والتمبليت تبع الأحمر نفس الشيء، أخذناه من الموجود already من الستراند الثانية. أوكي؟ صح، ممكن يكون الأليل مختلفين. لكن ايش احسن شيء السل ممكن تختاره؟ تصلح الدي ان اي ولا الاليلز يكونوا مختلفين؟ الاليلز يكونوا مختلفين، ما تصلح الدي ان اي ولا الاليلز يكونوا مختلفين؟ لا، بدنا تصلح الدي ان اي ولا الاليلز يكونوا مختلفين. eventually انا عندي الجين، الخلية ما خسرته، صحيح؟ اوكي. هذه واضح شوي احسن؟ يعني شوي احسن؟ فهمناها؟ يعني نص بدنا اياها 100%. طيب كذب علي. اوكي هشرح كمان مرة وكذب علي. اوكي so the idea انه انا عندي هلا 2 strands هون. عندي chromosome 1 بس. عندي 2 strands broken او صار فيهم deletion، صار فيهم cut whatever. وعندي بالchromosome 1 الثاني homologous chromosome عندي 2 strands. ماشي؟ So the idea since these are damaged هناخذ هناخذ good region من هذا الجود كروموسوم من واحدة من الستراندز هناخذها وحنحطها على الثاني. هلا صار هذه الستراند كويسة. It can be used as a template to fix the other strand. وهون well already واحدة كويسة والثانية أنا نشلتها الثانية اللي أنا نشلتها صار في gap. Well I can fill in the gap من الستراند المنيحة. أوكي؟ فهذا هو الهومولوجوس ريكومبينيشن، انه هومولوجوس لانه هومولوجوس كروموسومز، حطيناهم مع بعض واستخدمنا الهومولوجوس كروموسوم عشان نرائع. In other words. Got it? Okay. It turns out in breast cancer there is a gene called BRCA1, and there is a cousin, the BRCA1, known as BRCA2. These two genes are responsible for a good percentage of cases of breast cancer. وطلع إنه هذا الBRCA1 it's involved in homologous recombinational DNA repair. It makes sense. لما يكون عندي mutation بالBRCA1 معناته إنه if there is damage in DNA it cannot be fixed. معناته راح يصير عندي accumulation of mutations، راح يكون تتراكم the mutations and this can lead to cancer. Okay? فتقريبا 10 to 15% of the breast cancer cases are familial. يعني transmitted من grandmother للmother للدaughter. A good percentage of these cases they have mutations with BRCA1. Okay, so BRCA1 what it does إنه في شغلة بس هون إنه RAD51 هذا البروتين is involved in homologous recombinational repair. Okay, it turns out إنه the one function of BRCA1 it takes RAD51, this protein, it takes it to the site of DNA damage. فلو كان BRCA1 mutated or defective, معناته it will not be able to function, it cannot recruit, it cannot guide RAD51 the region of damage, معناته the DNA cannot be repaired. And it turns out that BRCA1 is also involved in transcription as well as coupling transcription and repair together. Okay? Questions? هلا حكينا احنا المره الماضيه على الميتوكوندريال دي ان اي، وحكينا على الكونتروفيرسي هذه كيف انه احنا ممكن يكون عندنا human being with 3 DNA molecules من المذر ومن الفاذر ومن دونر. Okay? هلا these are true cases والfield of molecular biology عماله بتحرك بشكل سريع جدا انه الناس المتخصصين بالاثكس مش ملاحقين على الاشياء اللي بتطلع لنا كل شوي، خصوصا انه عندنا دول ما بتهتم كثير بالاثكس. يعني الغرب بيهتم بقول لك لا في شيء اسمه اثكس، لكن عندك الصين مثلا، الصين is a free society بيعملوا اللي هم بدهم اياه، ما في كثير ما في high level of ethics عند الصينيين. كوريا تقريبا نفس الشيء. اوكي؟ وهاي الدول ممكن تعمل اللي هي بدها اياه، الجينيتك modification والengineering اللي هم بدهم اياه. هلا and scientists take advantage من اي شيء بيكتشفوه جديد. واحد من الشغلات اللي اكتشفوها جديد انه البكتيريا نفسها عندها genetic system بيشتغل كانه immune system بيحمي البكتيريا من الفيرال انفكشن. Okay, it's quite amazing. اسمه CRISPR Cas9. Cas9 هو endonuclease. الكريسبر هو السيستم هذا. اقرأوا عنه. It's quite amazing. هلا لما درسوا الكريسبر Cas9 أكثر اكتشفوا إنه it has the ability أو it can be utilized إنه إحنا نشيل fragment من ال DNA and replace it with another one. Okay? So think about it. لو واحد عنده لو هو carrier للسيكل سيل أنيميا وزوجته carrier للسيكل سيل أنيميا كمان، معناته there's 25% chance إنه الولد راح يكون affected with sickle cell anemia. بيعملوا in vitro fertilization. بنشوف بنعمل diagnosis أو نعمل DNA analysis وبنشوف إن الولد will be affected. عنده mutation على both genes, both chromosomes. فإحنا قلنا ليش ما نستخدم الكريسبر Cas9 وندخله على السيل ونخليه يروح على الجلوبين جين ويشيل الفراجمنت اللي فيها الميوتيشن ونحط بدالها DNA fragment ما فيها mutation. Can it be done? Yes, and it has been done. In humans? In humans. عملوها. طيب, is that ethical? Yes. ولا مش ethical? Sickle cell anemia. ethical. اوكي ethical. اجى واحد اتطورت التكنولوجي، اجى واحد حكى انا بدي ابني يكون عيونه زرق. اوكي؟ ethical ولا غير ethical؟ هو حر. بده ابنه هيك يطلع. ممكن حدا ينط يحكي انا ethical. ليش؟ لانه اتس اكسسوري، يعني مش شيء ضروري. Is it accessory؟ Yes. طب اذا اكسسوري ليش الكل خدوده طالعه ومشدود والبوتوكس بكل مكان؟ اوكي؟ لانه مهم. عند كثير ناس هذا شيء كثير مهم. لانه العيون الزرق والشعر الاشقر هو اللي بيفتح المجال للشغل وللنجاح بالحياه. صحيح؟ مهم. سوشيالي مهم. اوكي؟ I don't know. لكن I agree with you, it's unethical. طب افرض انه واحد بده يعمل genetic modification using CRISPR-Cas9 علشان يخلي ابنه اقوى، healthier. ethical ولا unethical؟ think about it. يعني يخلي الولد احسن. debatable. هون debatable, of course debatable. هون بندخل احنا بالجراي اريا. طب ايش الصح وايش الغلط؟ داخلين احنا بالمولكيولر بيولوجي بايشوز لها علاقه بهذا الحكي، يعني زي هذه الست اللي قررت انها بدها تدرس الزايجوت. اوكي؟ ففي ناس بيقولوا listen بما انه صار في fertilization it's a human being. خلص ما في مجال انك تتلاعب فيه، وفي ناس بقول لك لا على 41 و 42 يوم، في ناس بقول لك على 4 اشهر، في ناس بقول لك you can do whatever على اي عمر اللي بدك اياه. ماشي؟ بين الكالتشرز في اختلافات. هذه قررت انها بدها تتلاعب بالجينات using CRISPR-Cas9 بالزايجوت. ليش؟ الهدف تبعها سامي. هي بدها تفهم الجينات ودورهم بالديفلوبمنت والديفرنشيشن على اساس انه لو صار عندنا اي ميستيكس تفهم البروسيس البيولوجيكال بروسيس على اساس انها تضبط على اساس انها تضبط اذا كان عندنا ديفكت عشان تزيد من الشانسز تبعت الفيرتلايزيشن والاي بي اف. طب ايثيكال اللي هي بدها تعمله ولا ايثيكال؟ جراي اريا مره ثانيه. جراي اريا لك بس لكثير ناس نو وجههم بيحمر وبضربوك honestly. فاحنا داخلين بمجالات كثير لها دخل بالافكت. طب اعطيكم مثال ثاني، جينيتك تيستينج، حكينا على براكا 1 هلا. طيب are you willing to undergo genetic testing عشان تعرفوا اذا عندكم براكا 1 mutation؟ عشان تعرف اذا عندك براكا 1 mutation خصوصا الفيميلز. yes strong yes. طيب افرض صار عندك انه طلع عندك ميوتيشن بالبراكا 1. اوكي؟ يعني عندك تشانس مش 100% تشانس انه راح يصير عندك بريست كانسر. لا سمح الله لا سمح الله. اوكي؟ رايك في حدا بتجوزك؟ You don't care, you care.
I really don't.
You say you don't care لأنك you're young. Okay? Really في ناس مخهم بقول لك no. اوكي؟ هيك خلاص. اوكي؟ بقول لك لا، في عندها ميوتيشن، في احتمال يصير عندها بريست كانسر، احتمال انها تغلبني لما تكبر، whatever. في ناس هيك بيفكروا. مع انه مش 100%. مش 100% and it is treatable. بس في ناس يعني في عندنا سوشيال ستيغما، هاي العيله ما تقربوا عليها عندهم ميوتيشن. اوكي؟ طيب افرض تجوزتي وبدك تقدمي شغل والشركه عرفت انه عندك ميوتيشن بالبراكا 1.
كيف وصلتهم المعلومه؟
Why would they know؟ هلا بحكي لك how they would know. أنا وهذا بحكي لك. الشركة بدها تعرف ليش؟ لأنه احتمال تكلفيهم مصاري. شركة التأمين ممكن الشركة تعينك، التأمين ما يعطيك تأمين، يقول لك لا أنت راح تكلفيني مصاري. لأنه they think about money. Capitalism. اوكي؟ ما راح يعطوك تأمين صحي. How they would know؟ بحكي لك how would they know. لأنه people talk. وما في respect للprivacy ولا confidentiality. at all. بالمستشفى امشوا راح تشوفوا المفاعيس الريزيدنس هذول اوكي بالاسانسير. شفت فلان المريض لما فحصناه شو طلع عنده؟ انتوا بتحكوا بس هي it happens بيحكوا كثير. طيب او بالسوشيال جاذرينج دكتورين بيحكوا مع بعض واجا عندك المريض اللي بعثت لك اياه طب شو هذا شو رايك قدام الناس كلياتهم؟ الفحص تبعه طلع هيك والفحص تبعه هيك وطلع عنده ميوتيشن ولا وات ايفر. كيف فصار في سوشيال ستيغما لانه ما في ريسبكت للانديفيدوال ما في ريسبكت للبرايفسي والكونفدينشياليتي. فالنصيحه منكم really keep that in mind. Keep that in mind حكايه الكونفدينشياليتي and privacy and respect for others. يعني في كثير اشياء لها دخل بالمولكيولر بيولوجي we can talk about it later on. اي سؤال؟